Okay, we're going to start a little bit early, maybe get you guys out a little bit earlier for the keynote speaker. So, Dr. Burrows serves as the Director of the Office of Scientific Investigations within the U.S. Food and Drug Administration Center for Drug Evaluation and Research, Office of Compliance. In this role, Dr. Burrows leads efforts to shield the American public from unsafe and ineffective drug products. His leadership in the agency's Bioresearch monitoring program includes the development and implementation of policies, surveillance activities, and compliance strategies for CDER regulated products. Additionally, he oversees the enforcement of clinical and non-clinical drug product studies, bioequivalence studies, human subject protections, post-market adverse drug experience, reporting requirements, risk evaluation and mitigation strategies, and post-marketing requirements. Please welcome Dr. Burrows.
Thank you. It's a pleasure to be here. Really appreciate making the trip out. Never been to Ohio before. Went to a lot of college, saw a lot of universities. Thought I knew what a big school looked like. I wasn't even close. My perspective on that has shifted dramatically in the last 24 hours. Happy to see so many people. I really appreciate the opportunity to come to an event like this and talk with people at a university setting like this. Normally, the type of outreach that I do, it's a different crowd. It's all people that are in it for different reasons. So I think this is a really meaningful opportunity to talk with you all today. You heard who I am, where I am. It's a mouthful. I'm not going to repeat it. Topic for today is CDER BIMO compliance and enforcement, what you need to know. The reason I talk about compliance and enforcement is because those are two completely different things. First, compliance, what you need to do to be able to adhere with the regulations, and second, enforcement, what happens if you don't. I know normally you hear an FDA presentation and it's kind of like watching a dog walk on its hind legs. It's not particularly graceful or elegant, but you don't care because you're just impressed that they can do it at all. I tend to approach my talks like this a little bit differently. I think you might be able to tell that by now. I think we have a really nice show for you. My intent is to get you some valuable information in a usable format with enough humor to keep you interested, but not so much that makes you think I don't take it seriously, because I really do. And I think I'm probably going to have maybe 15, 20 minutes at the end for questions. So throughout the course, if you come up with anything that you think is going to be like the biggest, best question there is, please just keep track of that and see if you can stump me at the end. How's that sound? Doing okay? So the agenda today, I'm going to start with some basic background to make sure we're talking about the same thing the same way, to build on a foundation of shared understanding for what BIMO is and the types of programs that we're talking about today. Second, I'm going to run through clinical inspection summaries. That is, I think, probably a new term for you, but it is really meaningful in describing why the work that you do in clinical research matters so very much. Second, I think probably one of the most meaningful topics for you, strategies for building quality into clinical trials. I have some really good nuggets of information that I think are going to add value to you in your day-to-day work. And then a great section on responding to the 483. Not that you ever want to, but just in the event that you might have to, I'm going to give you some good pointers on what you might need to do there. Before I get too far here, I want to maybe just take a quick poll, show of hands. How many people know what BIMO is? Basic understanding, bioresearch monitoring. Okay, I'll cover that. How many people have ever been part of an inspection or had a study that they worked on inspected or anything like that? Okay. How many people have read the compliance programs, the CPGM, for clinical investigator inspections, sponsor inspections, or IRBs? Fantastic. How many people are terrified of getting a warning letter from me? There we are. So the things that we're going to talk about today are specifically designed to keep me from knowing your name. I always say, if I know your name, you're already in trouble. So with that, here we are, away we go. BIMO. Bioresearch monitoring is a portfolio of programs that my office oversees. That includes good clinical practice generally. Most people know what good clinical practice is. I think most of you probably do. It's also human subject protection. That's the IRB, RDRCs. That's a Radioactive Drug Research Committee. We are full of acronyms. If I just gloss over an acronym that you don't know, please just raise your hand there and I'll see if I can get into that. But BIMO also includes bioequivalence, bioavailability studies, GLP studies, and now, new and improved, it includes all of the post-market safety programs. Those are paid adverse event reporting, REMS, risk evaluation mitigation strategies, and PMRs, post-market requirements. So the BIMO program is huge. But none of that matters because the only things that really matter are these three program objectives. First, right, safety, and welfare of research subjects. Second, the accuracy, reliability, and integrity of study data. And third, to assess compliance with the rules and regulations. Notice, please, that those second and third bullets are different things because the accuracy, reliability, and integrity of clinical trial data is not necessarily driven by compliance with the regulations. And you'll hear a whole lot more about that in just a few minutes. The reason we're here really is because this industry is about shared commitments. It's about commitments for developing safe and effective medical products. It's about having confidence in the clinical trial process and knowing that if you do things right, you're going to have a clear path towards approval for your new product. Inspections, this is the 10,000-foot view. I think probably the group that we're most concerned with is clinical investigators and sponsors. I know at academic medical centers, often you see sponsor investigators. In other words, one individual that's taking on both roles. Cripplingly complex activity to undertake as a single individual. You see the highest rates of noncompliance with sponsor investigators, something you definitely need to be aware of. Also IRBs, RDRCs, I mentioned that. I don't think Ohio State has a radioactive drug research committee, but I could be wrong. There's only like 87 of those in the entire country, and they have to be licensed by FDA. Not going to talk about that today because it's probably pretty boring. So what happens in an inspection? Everything you need to know about what's going to happen during the course of the inspection, you can find in public documents called the compliance program. The CPGM, now it's called just a CP. The Clinical Investigator Compliance Program goes through the instructions for the Office of Regulatory Affairs investigators on how they should conduct an inspection, the types of things that they're looking for, the types of documents they're going to ask for. It is your user guide. It is the Office of Regulatory Affairs user guide, and it's all public. So if you weren't one of those people that raised your hand about reading a CP for the Clinical Investigator Inspection Program when we started, I highly recommend that as homework. Why? Okay, so why do we inspect? It's not just to make your life difficult. I swear there's a point. We inspect for three basic purposes. First, we have application-driven inspections. When my office receives a new drug application, it might have 100 or 1,000 clinical sites. We go through those and use a risk tool to figure out which ones we want to send inspection assignments to. And we might go to 3, 5, 10 clinical sites, depending on the size of the study, to be able to assess how that study was conducted in practice, in reality. Those are the inspections to verify the reliability and interpretability of the data in support of the application. We also have complaints and referrals. These are not fun inspections. We get maybe 800, 850 referrals a year. Those are referrals of noncompliance. If you are in your facility and you see somebody doing something that you think is horrible, they are putting people at risk, that they are making up data, you might be so inclined to report that type of conduct to me. 800 of them a year, and those are some of the most ridiculous stories you have ever heard. The worst of the worst, the bad of the bad. The baddest of the bad. One of those. But the complaints, we do about 100 inspections of for-cause inspections of clinical investigators a year, and they do have probably the highest rate of official action indicated of noncompliance. And then we have surveillance programs. Those are the IRBs, the institutional review boards, the RDRCs, GLP facilities, and some of those other programs. Okay, so that was a super fast overview of bioresearch monitoring, good clinical practice in our inspections. I'm gonna slow down a little bit now to make sure that I can get the information that's really gonna be most meaningful for you. This is where it starts to prick up your ears. It's the dreaded classification of an inspection, right? NAI, VAI, and OAI. In other words, bug bites, dog bites, and shark bites. You look at these classifications and you say, okay, objectionable conditions or practices. Serious objectionable conditions. What's the difference? What does that really mean? How are you going to know whether or not these are objectionable conditions or serious objectionable conditions? It doesn't really add any value. These definitions, I think, are pointless, which is why we're going back to the compliance program. The CPGM for clinical investigator inspections has a much more robust definition of what official action indicated actually means. When you read that CPGM, you'll see this is the foundation for all of the work that Office of Compliance does. When we get an EIR, an inspection report in-house, when we see a 483, when we're reading a 483 response, what we keep in mind as we go through all of that evidence, all of that information is what this definition says. I'm going to pause and go through it just so we can be clear about what we're talking about. You have repeated. All that means is more than once, more than one thing, more than one instance. Deliberate, you knew about it, or submission of false information. If we're talking about submission of false information, you have a whole nother set of things to worry about. Significant, serious, or numerous, scope, severity, pattern, these are all just yada, yada, yada words. What I'm pointing to here is a finding. My office makes a finding. I'm a lawyer, findings are fun. The findings that a subjects would be or have been, I guess I flipped that, have been or would be exposed to unreasonable and significant. Think about the observations that you might see in an FDA inspection. When you are putting together your 483 response, the response to my office, it might be a good idea to keep in mind this definition and frame the issues so you could discuss with us whether or not these objectionable conditions were really exposing subjects to unreasonable and significant risk of illness or injury. Then subjects' rights have been or would be seriously compromised. That's a great discussion point too. What does it mean to seriously compromise subjects' rights? What level of compromisation is appropriate? And then lastly, another key distinction, data integrity or reliability has been compromised. Notice there's no qualifier there. Compromising data integrity or reliability, period. Automatically OAI. We'll put a pin in that and come back to it in two sections when we're talking about building quality and responding to FDA. So that's our definition. This is going to be a foundation. We're going to talk about that a whole lot more once we get to responding to FDA, responding to 43s. Moving right along. Background, clinical inspection summaries. I want to stop just for a second and talk about what my office does. My office is in the Office of Compliance, but we hold two roles. The reason we have two roles is really in our mission of protecting the public health. The first role, obviously, compliance. We enforce violations of the laws and regulations, but that is driven by what's actually required. Protocols are the blueprint for quality, and if you have a crappy blueprint, you're going to have poor quality trials. If the phrasing in the protocol has words like may or should, that's not required. It's not a violation. If the protocol has must and shall, that is a requirement. So the phrasing of the protocol itself will drive whether or not my office is going to see a violation in the inspection. Same thing with the regulations. The regulations were most of them written in 1988. It's been a hot minute. The system has evolved a lot since 1988. So the phrasing of the regulations is not always exactly on point to the operational realities that we see in clinical research today. Technical limitations, the data sources, the way that we collect, view, share data has changed an incredible amount since 1988. And industry contracting. A great example here, sponsors can contract out any or all of their responsibilities to other vendors, to CROs. And when they do that, they don't have any compliance responsibilities. We can't enforce violations on a sponsor when they have contracted out all of their responsibilities to another party. But that doesn't mean that those inspections still don't add value for the second part of our role here, the application review. Completely different. Think about it this way. If my office did inspections of clinical sites and found some widely discrepant practices among sites that called into question the generalizability of the data generated from that study. If the protocol was poorly written, we might not be able to enforce those violations towards the clinical investigator. But that wildly variable conduct across clinical sites is going to have a meaningful impact in our review of the application itself. Nobody wants drugs to be approved and provided to the American public where the data that shows that they actually work or are safe is unreliable. When we make our assessment of the reliability and interpretability of the data submitted in an application, I don't care where that information comes from or how bad the protocol was. If it's something that is material to the review of the safety and efficacy data, we're going to consider that in our recommendations to the review division. And the way that those recommendations show up is in a clinical inspection summary. Pause. How's that sitting with you so far? I'm going quick, a lot of information, doing okay. So a clinical inspection summary is one of the very most meaningful things that my office does. We have about 150 CISs each year going to the review division. And 2 years ago, I asked myself, how do we show what type of impact we are having with our CISs to the review division? What kind of recommendations are we actually giving them? And what's the end result of those? So we did an analysis. I'll pause for a CIS. So a CIS it is the report that compiles all the results from all the inspections we did for an application. So it could have 3, 5, 10 clinical sites. It could have an inspection of the sponsor. It could have an inspection of a CRO or a vendor of another system. Wherever we need to go to get the data that we need in order to feel comfortable with the study endpoints, the safety and efficacy endpoints, that's what we're going to do. And we compile all of those documents, all of that review, all of that analysis in one document. It's a publicly posted document at the end of a review. When an application's approved, you can see a CIS online. So it looks at all of those things. We put together a recommendation, all focused on data integrity. These are things that I'm sure you've seen and heard before. We're talking about the principles of ALCOA, establishing the reliability and the interpretability of data, and now becoming more and more prevalent is traceability. Now that we have new electronic data captures, ECRFs, we're using medical health electronic health records, different direct streaming documents and data to sponsors, vendors, and CROs. We need to be able to trace how a data makes it to us from its original source all the way to the NDA, to the application that we're reviewing. And also reliability, that is whether or not the data are reliable. Did XYZ actually happen? Interpretability is a little bit different. I touched on that a minute ago when I talked about the variability of conduct across clinical sites. If you have a provision in a protocol that says should, and half of the clinical sites interpret that provision to mean let's do it because we should, and half of the sites interpret that to be like it's optional because they just said should, then when we pool that data, is that really interpretable? Is it going to be true and accurate representation of what the intent of that study was. If that provision is something that has a material impact on the endpoint of the study, it needs to be done consistently across sites to be able to be interpretable. And if you have a poorly written protocol, it is probably not going to be interpretable. So what we did was we had to come up with a way to grade our recommendations to the review division, and I opted for the simplest way that made sense to me. That's the difference between an active recommendation and a passive recommendation. A passive recommendation is basically just saying, you're good to go. We looked at these 5 inspection sites. We didn't find anything that really called into question the reliability of the data. I think it's appropriate to support the application. An active recommendation is a little less nice. It's saying, we did these inspections, these are the things we found, we recommend that you do something. We're recommending to the Office of New Drugs that maybe they run a sensitivity analysis. Maybe we recommend actually excluding data from 1 or more clinical sites. We might need to do additional inspections to see if one thing that we saw at one site was an isolated instance or if it was something that was more systemic in nature. We might need to do more inspections to figure that out. We could do information requests. We could send an information request to the sponsor asking them to explain to us what they did when they found this issue in their monitoring of the study. So big buckets, passive recommendations, great, you're good to go. Active, you might need to do something. We looked at over 300 CISs over a 3-year period. It was probably 4,000 pages of text, and I had one person doing it because I wanted consistency in the way that that person viewed what they were seeing in the recommendations. I didn't want half of them to be reviewed in one lens and half to be reviewed in a different. So one person read the equivalent of all of the Harry Potter books and all of the Lord of the Rings books all at once inside of a year, which is a lot when you actually have to pay attention to what you're reading. But okay, so, ta-da! No, okay, so we found that 19% of our clinical inspection summaries had an active recommendation. Much higher percentage of active recommendations than we really thought we were going to see. The good news is there that in 81% of the applications that get submitted to the agency where we do BIMO, GCP inspections, we say the data are good. No questions, they're appropriate to consider in support of the application. In 19% of those cases, we say there's something that we want you to do before you just consider that data reliable. Not saying that these applications didn't get approved, not saying that they didn't get approved in their original user fee action date timeframe. It's just that there was a question that we needed to have resolved before we could provide our full endorsement for the data. Here's a key aspect that draws the distinction between those 2 roles that I talked about in my office holding. First, in compliance, we're looking at OAIs and what type of actions we take there. And the second is in the application review component. With the active recommendations, where we told the review division that they actually needed to look at something further before they approved the drug, almost all of those active recommendations were driven by findings from NAI and VAI inspections. That means even if you get an NAI inspection, FDA comes, looks at your study, says you're good to go, no actions necessary, it still could be true that the information collected from that one inspection is going to be pooled with information from other inspections, and we're going to say to the review division they need to take an action before they can consider that data to be reliable. When I first presented this data at DIA, not last year, the year before, maybe, it was a blind side to industry. They had never considered that NAI and VAI inspections can have this significant an impact on their applications. It is strictly the nature of the beast, and that message about the protocol being the blueprint for quality. If you have a poorly written protocol, we're not going to be able to hold clinical investigators accountable for the must versus the could, the may versus the should or the shall. So those NAI and VAI inspections can have tremendous impact on the review of the application as a whole. And those the active recommendations, really the most common one we saw was sensitivity analysis, seeing what the impact on the endpoint would be if you took out one or more sites. We also did a lot of additional inspections to pull the thread on things that we thought might be isolated to make sure that they were not systemic in nature. And the biggest component of this giant nebulous other category is the information requests to sponsors. If we found something, we just wanted a sponsor to explain to us what they thought when they saw this show up in their monitoring, what they did about it, and what the relative impact might be on the application themselves. We also found that when we had an active recommendation to the review division, when we had questions about the data, in 26% of the time, it was more than one thing that we were recommending. That might show up like excluding one site and recommending a sensitivity analysis for others. It could be that we are recommending an information request to the sponsor to figure out what happened with a given situation, and also a sensitivity analysis to see if you remove a certain cross-section of subjects, do you still meet the endpoint for the study? And that matters because when the data are reliable and interpretable, FDA can take prompt, decisive action on the underlying application. The end goal there is to get valuable therapies to market to people that need them. And when we have questions about the reliability and interpretability of the data, we can delay the review of that application. So keep in mind the value of the NAI and VAI inspections. Tremendous value, not widely recognized that those findings actually make their way into our recommendations more often than not. All right. Building quality into clinical research. It's a complex process. It's not something that happens in isolation. You can't just be concerned with your role and your role only. It is an entire system. And looking at the size of this academic medical center, you can tell there are people in one building doing things that people in other buildings might not know about. We did a study with CDER, with the Center for, Clinical Trial Translational Research. I messed that up, but CDER, an organization that we have a relationship with. They did, I wish this wasn't being recorded now. We did a study to see why investigators would just do one study and then quit. They never did additional studies. So often we see clinical investigators that sign up to be a clinical investigator, they do one study, they have a poor experience, and they never do it again. On the other hand, you have a much smaller cross-section of people that get involved in clinical research, and that becomes a critical part of their business, a critical part of their practice. They take to it like a duck to water. And the thing that we found that was the most critical differentiating criteria between the one-and-done CIs and the core business process people were that the people that engaged in clinical research and were successful had a quality system around them. They had a support network. The people that they were working with at their facility day in and day out knew that they were engaged in clinical research. They knew what their responsibilities were. There were systems in place to highlight errors when they happened and prevent them from becoming any worse. The individuals that just did one study and then quit were alone on an island. They were acting on their own. They didn't have friends and family that they could call. I'm using that in the wrong context, but they didn't have a network of people that they could call on in their facility to help them. Their research coordinators, if they had a research coordinator, weren't really in tune with what the requirements were, what the protocol said. There is a wide spectrum, a great continuum of relative sophistication in clinical research, from people that are sponsor investigators on their own, acting on their own, it's just them against the world, to people whose entire organization is dedicated to clinical research. And there are so many systems in place that even if that one individual tried to mess something up, they probably couldn't. It's a complex process, a ridiculously involved system. And we are just trying to peel away one little layer about that right now to explain some strategies that you could use to build quality into a system one step at a time over time. This is a great chart, plots risk on two axes. Xi plots risk two ways. We have probability of occurrence of a given risk and severity. The name of the game is taking the risks that matter and moving them to the bottom left. And there are strategies all throughout your organization that you can implement that will do just that. Sometimes you can take action that are going to do both at once, decrease the probability and decrease the severity. But when you're thinking about building quality into clinical research, you're talking simply about identifying the risks that matter and finding a way to move them to the bottom left here. So quality risk management, it's a systems approach. You know the quote, life's a journey, not a destination. Well, quality's a journey, not a destination. And you could attribute that to either Ralph Waldo Emerson or to Aerosmith. You get to take your pick, but quality is a journey, not a destination. You're always going to have errors. In any research practice with any protocol, you're always going to have errors. The name of the game is taking the errors that matter and making them less prevalent and less severe. Now, every organization, every clinical research practice is going to fall somewhere on this risk management maturity chart. I'd like you to pause for a second and think about where your practice, where your organization might fall. On the far left, you have the no care culture. It just doesn't matter. I didn't sign up for this. That's not a great place to be, but it happens. Next up, the blame culture. It wasn't me. I did what I was supposed to do. It was you. It was your fault. Also, not a great place to be. Then you have the middle, the compliance culture. That is perfectly acceptable. It's not a bad place to be. The compliance culture prevents accidents before they occur. You do a risk assessment. You have a causal investigation. It's not bad, but it is not nearly as good as what could be better. That is one step over, the ownership culture, where you're tuning systems through mistakes. You're learning organization that knows throughout your organization that quality, compliance, that culture of quality, that culture of compliance is your end game. It's a great place to be, but still, the ownership culture can just be in your isolated organization. There is a way, there are companies, there are businesses, there are practices where compliance and quality is a way of life. It is not about any individual department, any individual disease, state, or condition. There is no aspect of that organization that is given any preferential treatment. The whole organization knows that this culture of quality is paramount for their mission. That's risk management at the core of the business culture, not just a division, the entire organization. That is a great place to be and very difficult to get to. So I'm not going to ask you where you think your organization falls on this chart, but think about it. My suggestions of ways to move to the right of this chart are not great leaps. They are small steps over the course of time. Big change happens one small change at a time. If you come back to your staff meeting and say, we're going to be way of life, we're going to make this part of our business culture, that's going to be met with a resounding thud. It doesn't happen that way, just one step at a time. So OSI's focus. Our focus is on building quality into clinical research. It is not about inspecting quality in. You don't want my office to come to inspect you as a clinical investigator, find critical observations, to send you a warning letter, to come back out next year to do a re-inspection where we find more findings and give you another warning letter, or worse, disqualify you. That is it's the kidney stone of regulatory oversight. It is the most painful way possible to get quality. I highly advise you to build quality into your clinical research practice from the beginning, not letting me inspect quality into the practice. So I've said this once before or twice before, but I'll say it again. The protocol is the blueprint for quality. If you have a clear, well-articulated protocol, the level of understanding across your organization is going to increase. Your ability to meet those commitments is going to increase. The name of the game there is making and managing a reliable network of commitments in your organization. So every person in the process knows what their responsibility is, what their colleagues' responsibility is, what the CI's responsibility is. They know because it's clear in the protocol. Yeah, okay, moving on. So, a horrible analogy that I'm going to use because it makes a great point. Building quality into clinical trials. What we do is we start with the foundation here. That is the risk assessment. Before you start a study, conduct a risk assessment to figure out what the areas of quality for data, for human subject protection really are. What is it that matters more than anything else? How do you use the quality by design principles to put together a study that's going to get you the end result that you're looking for in the easiest way possible? From that first risk assessment, that's when you build the livable space, the protocol. The protocol has to be built on a clear risk assessment. It has to be tailored to the way that you have structured your risk assessment. If you do that, you're on a solid foundation, you're doing really well. On the top here, we have the monitoring plan. You cannot take just any monitoring plan and apply that to just any study. A risk-based monitoring plan needs to be based on the risks that you found in your original risk assessment that you used to drive your protocol, to build your protocol, and that monitoring plan goes on top of that. It is a system that has to go together. These three parts need to go together. You can't just say, I'm going to use this protocol that I used last year for this other study and just change the endpoint and then add on some other lab values, add on some other tests, and then call that that. What we call that is a Christmas tree protocol. Have anybody heard that term before? A Christmas tree protocol is exactly what I just said. You just take it out of the attic, you dust it off, you add a few more things, and then over the course of years, you have a horrible protocol. It happens all the time. Why? Because it's always easier to start with what you did last time. You start with a protocol that you've used in the past and maybe had some success with, and then you just tweak it. You just add some stuff. You don't ever take away, right? It's not like you look at your Christmas tree and say, I don't need this ornament anymore. Like, oh, my daughter made it in 5th grade. Of course it's going to stay there. Christmas tree protocols are horrible. And that is how you get a protocol that is inconsistent within itself. A Christmas tree protocol is one where it will say you must do XYZ in one section and you may do XYZ in another section for the exact same thing. When you have a protocol that is inconsistent within itself, nothing in that protocol, none of those provisions can be violations. Because how could I ever tell you that you violated a protocol if you complied with one section and in doing that violated another part? And soapbox off. So, building quality into clinical trials, getting back on point, risk assessment, protocol, monitoring plan, all goes together. Now, I have 4 steps that you can use to build quality into your clinical research system. First one here, I think I've beaten that horse to death. It's create a quality system, right? The best point that I can make here is about bullet number 2. Create systems that increase visibility of errors. That's a fun one. As soon as you make it so that an error becomes immediately visible in real time, magically, it goes away. It is like magic. If you make a system where you can't hide mistakes anymore, the errors just disappear. If it's true that, that necessity is the mother of all invention, then it is also true that humiliation is its father. If every mistake you make is highly visible immediately, you're gonna stop making those mistakes. The best way to increase the quality of your system immediately is to find a way to make the errors that occur visible immediately. Magic, they go away. Next, select qualified staff and ensure that they receive adequate training. This is one that it always resonates really well with some of the conferences that you speak at where they have clinical research staff, the CRAs, the CRCs, the study coordinators. Because the point is that when you operate in this constant state of overwhelm, you increase the chance for error. You need to be able to be mindful of the relative workload that you and your staff have. If you are a two-person department and you sign up for 20 clinical studies at once, you're going to have a bad time. It doesn't matter how qualified the staff you have are if they are critically overworked all the time. So qualified staff, what does that mean? That means that you know your role, you know their role, you know what their capabilities are, what their strengths are, what their relative workload are. You get them the training that they need to be able to succeed in the tasks that you have assigned them. And this last point I think is critical, probably for this audience, but I won't bank on it. That's the difference between a clinician and a clinical investigator. How many people have seen House, the TV show Dr. House? Dr. House would be the worst clinical investigator in the history of clinical investigations. He would be a horrible clinical investigator because to be a clinical investigator, you have to follow protocol. You have to know what the rules are and actually adhere to them. There is a significant difference between the skill set that makes a practicing clinician exceptional at their job and what makes a clinical investigator exceptional at theirs. That is not to say that you can't have a great clinician who is also a great clinical investigator. It just means you have to be mindful of your role and where you are in practice at that time. This always strikes physicians as being a little hard because it's like saying, this skill set that made you awesome is going to make you horrible at this. I don't know if you really want to do it. It's not the truth, right? These are really intelligent people. You just have to be mindful of what you're doing when you're doing it, and the fact that a clinical investigator needs to be able to follow a set protocol, set regulations. It's not Wild West. It's not do whatever you feel like you might be able to do in order to help the patient right now, now. There's a protocol. Ensure adequate training. Training happens really 3 different phases, right? There's just 2 bullets here, but there's 3 different phases. You have training of your staff before you start a trial. Seems pretty intuitive, but doesn't always happen. Train before you start. Always a key for success. Second is train when people change. When you have new people come in to your organization to work on a trial midway through, you need to retrain everyone so you don't have the situation where it's like me, you, me. Everybody needs to be trained on the same protocol, knowing that they have made this shared commitment between themselves and the organization. And lastly, change when the expectations change. When you get a protocol amendment that changes what you need to do under the protocol, you need to retrain all of your staff on it. Probably not going to be effective to send a memo out, to send an email to all staff saying, oh, by the way, we have to do X, Y, Z different. You need to be able to train your staff on what changed and document the fact that people attended that training. So train again, test on it. Next up, fully understand the scope of your responsibilities. That it seems like it should be pretty obvious, but it's not. We have a lot of situations in practice where the clinical investigator signs a 1572, they sign up to be a clinical investigator, and they have no real role in the operations of that clinical trial. They expect everybody else to do everything. You have a sub-investigator that signs off on anything. In those instances, your clinical investigator is basically just expecting their name to be first on a paper, and that's it. A clinical investigator needs to fully understand the scope of their responsibilities. When I come and do an inspection of a clinical investigator, it's not the sub-investigator that I'm going to send a warning letter to. It's the most responsible person for that study. It is the clinical investigator. They need to know what their responsibilities are. And here, number 4, implement systems to detect and correct errors in real time. The biggest and the best, I talked about that before. make errors more visible, and magically they just disappear. Now, the best part of the whole presentation, how to respond to the FDA. It says respond to Form 43s, but it can be any response to the FDA. Pause. What I'm talking about here is not how to fool the FDA. That is not this. What I'm talking about is how to frame the issues in the light most favorable to you. It is taking the truth and putting it on a silver platter so that reviewers in my office can say, that person knows what they're doing, I totally understand, I agree, NAI. It happens all the time. In order to have that type of review, you need to be able to respond to your 483 effectively. And I'm going to go through every bit of what you need in order to be able to do that right now. I see that we have 20 minutes. This isn't going to take 20 minutes, I promise. I've broken this down into 3 sections. First is just some basics about responding to FDA. Second, 4 reasons to submit your complete, well-reasoned response to FDA, and 9 suggestions about how to make a response as impactful as possible. So, response basics. It is not a requirement that you respond to the FDA 483, but it is absolutely 100%, 110% if that was a thing. It's in your best interest to do so. Here, remember that a 483, an FDA 483, the list of observations from an inspection is just that, a list of observations. It is not a conclusion. The things on a 483 are not necessarily final agency determinations. The bioresearch monitoring programs are set up in a very unique way in FDA. They're set up so that the inspectorate, the Office of Regulatory Affairs investigators, do the inspection, write up a report, and they send it to my office in the center for a final review and classification. They don't make the decisions about whether or not something is significant or not. They don't make the decision about whether or not the inspection is OAI or not. And the reason the whole program is set up that way is because you have to have the scientific and medical expertise to understand what is and is not significant in order to make the decision about what's OAI. Remember that, that OAI definition from earlier in the presentation: scope, significance, significant impact on the right, safety, and welfare, on the data reliability, data integrity. All of those decisions are made at the center. This is a great opportunity for inspected entities, because what that means is if you get a 483 at your facility and you have 15 days to write that response and submit it, my office doesn't get the inspection report for 30 days. That means that your response can be the very first thing that my reviewers look at when they get a new case in. You have the opportunity to frame the issues in the light most favorable to you. When I was a reviewer, I always looked at the 483 response first. I instruct all of my reviewers to do that because of all the people who have a vested interest in being clear and correct in their analysis of your 483, it's you. Remember that the ORA investigator is at your facility for maybe a week. You live there. You know where the records are. You have the ability to take a deep dive into every bit of information at your disposal. They don't. They take what they can get in a week or so and then make their conclusions based on that. So remember that the 483 is just a list of observations, not conclusions about compliance. Next up, this is a statement from the IOM. That's the Investigators Operations Manual, not the Institute of Medicine. It says that investigators should make every reasonable effort to discuss observations with management in real time or at the end of each day. That means that in that conversation during an inspection, you have the ability to say, oh, I see what you're looking at here. I think we actually have some information that might add value to that analysis. Maybe we have these lab values in our medical records. They're just not in the study records. Let me go get them. And then you can undercut those observations. You can prove them to be false in real time during the inspection. I wouldn't recommend printing out that section of the IOM and chasing down your ORA investigator and say, you have to meet with me right now. probably not going to be in your best interest. But keep in mind that this is a general instruction to the ORA investigators. They should be talking with you about the things that they're seeing in real time. Now, 4 reasons. This is my favorite slide. I'm going to share with you information that I never put in slides because it's always best to not have it in slides. A complete and timely response could change the classification of your inspection, and it could change the type of letter that we send to you. It could make the inspection not OAI, but VAI. It could make it an untitled letter instead of a warning letter, an untitled letter being just to you and not posted on the web. We avoid public shaming. What you need to do in order to make your best case, to frame the issues in the light most favorable to you, is to include 3 things in every response to every observation and every instance within each observation. Those 3 things are these. The first, assess whether it is true. The ORA investigator is at your facility for just a week. You have access to all of your records all the time. Often, we find observations on 43s that we find later are not factually accurate. They are not correct. Some of the ways that might show up is, like I mentioned before, you might have records in your medical history for a patient of a blood draw, lab values that were required by the study, but the copy of those lab values are not in the study records. If ORA, if the investigators are there and they just get a copy of all your study records and they see that those lab values aren't there, they will cite you for it. But it's not true. It's not factually accurate because you know you have the record somewhere else. When you go through your 483, after the ORA investigator leaves, assess every single observation in every single instance to figure out if you have information that contradicts that. It happens often. If you find information that contradicts the validity of that factual observation, give us that documentation, explain what happened, where it was, and that observation will fall off. We won't reissue a 43, but our reviewers will know that it is not to be considered in the assessment of the inspection overall. All right, so first thing, factually accurate. Are the observations true? For those observations that are actually true, second question, was it really a requirement? That's when you go through the protocol and look to see if the phrases were should, would, was it must, can't, may, shall. Some of those mean you have to do this. Some of those mean, it might be nice. If they're using may or should, if they're using those types of phrases and they're listing that as a protocol deviation, that's not a requirement because it says may, not must. If you find that, make a note of it. Say this is not a regulatory requirement because the protocol said may, not must. Also shows up in those Christmas tree protocols where you have competing provisions, where the same thing for visit 1 might be phrased as a must in one section of the protocol and a may in another section of the protocol. When that happens, it is not a violation because we can't expect you to comply with one part while actually violating the other. So, for every observation, for every instance, is it factually accurate? Is it a true reg requirement? Third, significance. Remember that OAI definition? This is your opportunity to explain to our reviewers why whatever happened, if it's true and a reg violation, why does it not matter? Why is it not clinically significant? Why does it not have a significant impact on the human subject protection concerns of that study for those subjects? It happens all the time. You have labs that aren't material for the endpoint analysis or for any safety concerns. You have ECGs just as a matter of course because everybody has to do an ECG all the time even though it has no impact on the drug, could never have a safety signal generated from those ECGs. It's just a legacy from a protocol that somebody chose to build upon. Look at the clinical significance. What is the standard of care for that disease state? What is the relative mechanism of action? How sick or healthy are the subjects? Explain the significance. Why is it or is it not clinically significant? Why is it material? If you do that in your 43 responses, if you can frame your analysis saying true or not true, violation or not violation, and from all the things that boil down through that, is it significant or not significant and why? You will have framed the issues in the light most favorable to you. Our reviewers will love you for it because you will have just done their job. I absolutely recommend that if any of you find yourself responding to a 43, you follow that paradigm. Absolutely in your best interest. Next up, not nearly as in-depth, 4 reasons to complete a timely written response: demonstrate acknowledgement and understanding. I know that a lot of the research that you do in practice has significant scientific value. Your response to the FDA should not argue that your research is so important you should get a free pass for violating the regs. I'm not going to really say I don't care about the scientific value of your research, but I don't really care about the scientific value of your research. We are the Office of Compliance. We care whether you follow the protocol or not. More often than not, you see physicians that are writing their own responses to the 43 and arguing in support of their research, not arguing what happened, not talking about the observations that we found during the inspection. So demonstrate your acknowledgement and understanding. Second, demonstrate your commitment to correct the issues. You often see responses to 43s that are drafted by the legal department of your institution, and in many cases, the clinical investigator that we inspected doesn't even sign the response. If I am concerned about the clinical investigator's ability to prevent the recurrence of this or similar observations in the future, and that clinical investigator doesn't sign the response, that means nothing to me. I need to be able to see personal accountability and ownership from the directly responsible individual involved. That doesn't mean that legal can't draft the response for you, and it doesn't mean that they can't help you with it, and it doesn't mean that they can't sign it. It just means that you need to be there too. And establishes credibility with the FDA. What I mean by that is I love the responses where it says, we acknowledge this happened. It's, for reasons 1, 2, and 3, not significant. We've taken efforts to prevent its recurrence. Those include 1, 2, and 3, XYZ. We trained all of our staff on this new procedure. I've attached the training. I've attached the sign-in log from the training. We plan to do an internal audit in 3 months to assess its efficacy in preventing the recurrence of this violation in the future, and we'll follow up with a report of that analysis after the 3 months. That's a great response because it shows credibility, demonstrates ownership. Sadly, those type of responses are few and far between. Again, you get the opportunity to frame your response in the light most favorable to you. You get to put your stamp on the reviewer's analysis of your case before they actually even get it in-house. That can be the first thing. I look at this and I say, it's evil, of course, because I'm a lawyer. Dirty, blood-sucking lawyers. But you get to frame your perspective. If you convince somebody that this says good, 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 of course it says good, it says good, it says good. All you're going to see is good. I'm going to say good one more time. So frame the perspective. That's your 43 response. Now, 9 suggestions for a response. Include a commitment from senior leadership. You have big institutions. We know who controls the resources that are deployed to create quality systems. Include that commitment from senior leadership, making it clear that they are owning their role in this system. Address each observation separately. Remember, true in fact, reg violation, and significance, those 3 things. Note whether you agree or disagree. If you disagree, please provide the documentation that serves as the basis for that conclusion. Give me the evidence to be able to verify that that is in fact true. Provide both corrective and preventive actions. If it's something that you can fix, fix it. If it's not, prevent it from happening again. You have to respond within 15 days. I know that that means that you can't complete all of your corrective and preventive actions in 15 days. So talk about planned actions also, and include that timeline for completion. What's the commitment for following up once you've finished? Method of verification, I talked about that already. If you're going to do an audit in 3 months to assess whether or not that change was effective, make that commitment in your response and share that audit report with us once it's done. Submit all the documents that you've created in response to those 43 observations, and please, please, please respond within 15 days. Now, what if you miss the 15-day deadline? That means you just get this. This is a statement that shows up in our warning letters if your response was late. It means that we don't give you a blow-by-blow analysis of what was right or wrong with your response. It just says it was after the 15-day window. So, a couple examples of an inadequate response. I would highly recommend not doing this. Failure to obtain informed consent properly, general observation. The response, as to the falsified signatures of 4 out of 80 patients, suffice it to say we're talking about a margin of error of 5%. This is well within recognized statistical limits. If you don't already know, there are no statistical limits for fraud. Next, failure of the clinical investigator to obtain informed consent, follow the study protocol, and comply with commitments of FDA regulated research. That's just all of the observations that were on the 43. Response here, I signed the investigator agreement, but I never read it. During a real estate settlement, do you read all those documents before signing them? I'm not your attorney, but yes, yes you do. Another one, failure to assure that IRB was responsible for initial and continuing review of the proposed clinical study. The response here, I put together an IRB of regular folks to act as the IRB. The people that you meet in line at Starbucks are not your IRB. The written response was way worse. The IRB used to provide oversight of the study was a group of individuals I assembled to act as the IRB. Okay. I do not have any background information on the individuals that acted as IRB members. Doesn't even know who they are. I don't have any IRB study files to include IRB approval, IRB approved informed consent, or IRB progress reports. Absolutely nothing. It's not a great response. Doesn't make me feel warm and fuzzy. This person was disqualified. So that happened. So now, it wouldn't be good if I gave you all the bad responses, but not a good one. This is a great response. It's a template that you can use for just about any observation, as long as you still follow those 3 things that I included, the facts, the regs, and the significance. Failure to follow the investigational plan, here's the response. Prepared and adopted a written procedure that will help staff to assure compliance with written study protocols and the obligations we accept as clinical investigators for FDA-regulated trials. Acknowledgement, understand. A copy of the approved SOP is attached. There, documentation, great. We've reviewed the new procedure at a meeting held on whatever date with all research staff, attendance sign-in sheet attached. They can prove it. Implemented on XYZ date. You have a clear start date. After 3 months, we will evaluate these new practices to determine if the corrective actions assist in protocol adherence. The only thing you could add to this is the commitment to send that report to FDA after you did that 3-month review. That's a great response. It could be copied in just about any observation. It doesn't have to be 1,000 pages. It can be clear, concise, and right to the point. And I've gone just about over time. So what we talked about, mechanisms to build quality into clinical research. Please, if you remember nothing else, the protocol is the blueprint for quality. A crappy protocol will get you a crappy study. Strategies for responding to FDA. Please, factual accuracy, regulatory applicability, and significance. And these are some resources, but in true FDA fashion, we just changed our website, so these links no longer work. If you need anything, just Google it and it'll come up, right? And that's it for me. And I went over. Thank you. So you got me into monologuing a little bit there. I'm sure not on purpose, but I used up my full hour. I'm happy to take any questions now, and I will stick around for a Q&A period afterwards if that is at all interesting to you. Any questions at all? Going once, going twice. Go ahead.
So a little bit of conflict in describe how quality is so important. Protocol is so important to have a clear and concise. But then on the other end, if it's a little wishy-washy, it might save you at the end of the day if you have some May language in there. So
Yep.
is there a balance? Is it one or the other?
You know, there's always a balance in drafting a protocol because you can make a protocol that is clear and specific and directly on point, but not really operationally feasible. But the interest is in having reliable data. And when you have those squishy protocols, it's going to be unreliable data. And the reason for that is this question of interpretability. If you're using a protocol that has all the squishy language in it across multiple sites, I know that whatever that data point is unreliable because it wasn't done consistently. It was a should and not a must. So yes, in theory, but the value of the research then takes a significant knock. And side note, to the extent any of your research is published in a peer review article, they will be able to see what was in your protocol and be able to name that as a limitation. Please.
Great talk. Appreciate all the feedback. This has been all folks around GCPs. I respect that. Does your office also do you mentioned GLPs, manufacturing as well?
Not manufacturing, completely different office.
Completely different office. Okay. Which office is it?
That's the Office of Manufacturing Quality.
Okay.
If you would like a contact, I could certainly get you one.
Thank you.
I'm lucky I can spell manufacturing. That's as much as I know about it. Anything else? Excellent. Well, I'll stick around. Anything you want to ask off the record or otherwise, please just feel free.